Welcome to the Vermont Center on Behavior and Health's December 2022 lecture in our monthly lecture series. And I'm thrilled today to have Alexi Potter with us, a professor of psychiatry in UVM, University of Vermont, Larner College of Medicine. Alexi completed her academic training here at the University of Vermont. She is green and gold through and through, completing her bachelor's degree, her doctorate in clinical psychology, and her postdoctoral training, all here at UVM. And I've been here long enough to actually have been around Alexi in all those different stages. Her research focuses on using neuroimaging, experimental manipulations, and pharmacological challenge to understand individual differences in cognition and behavior. She's kind of has, as many of us have, different stages in her career. So her early career work focused on the role of the nicotinic acetylcholine receptor in impulsivity, and then as a medication response or site for ADHD medications. But over time, her interest broadened, her interest in the neurobiology of psychiatric conditions broadened, and came to include large-scale, multi-method research to understand individual differences and vulnerability to psychiatric conditions, and led to her involvement in NIDA's Adolescent Brain and Cognitive Development Study. It's a national longitudinal study focused on addiction vulnerability, but also is collecting data to allow you to really understand a wide range of different vulnerabilities. And that's the topic that Alexi is going to be talking about today. As you can see, her title is Insights About Substance Use from the Developmental Neuroimaging Studies, an update on the ABCD and the HBCD studies. So this is really the largest scale work of this nature ever done, as far as I know. And I'm thrilled and proud that UVM has a part in this work through Alexi and her collaborator, Hugh Garaman. I meant to acknowledge her collaborators in the early stages of her career, which was Paul Newhouse and Julie Dumas. Paul has moved on to Vanderbilt University, and Julie is still with us doing outstanding work along the lines I mentioned regarding Alexi's early career. So the work that Alexi and Hugh Garavan are doing now is just terrific. They started out, as I understand it, being the coordinator center, but then they evolved where they were one of the research sites for this multi-site study. They've grown it in different directions, looking at opioid use disorder and all kinds of different things I'm sure she's going to tell us about. So without using up more, I did want to say one other thing. Alexi wears multiple hats, and one of the things she's done in way of service is she has chaired the Institutional Review Board for the Medical Sciences here at UVM. And I know that our center has benefited immensely from that. And then she also is a collaborator with the Vermont Center on Behavior and Health, helping us assess executive function, in particular, with a wide range of different cognitive and behavioral skills. So without further ado, I give you Alexi.
Thank you, Steve. Be good one. This one. Can you hear me through this microphone? Are we on? Yes. No. Yes. I get both the answers. Ish. Here, I'll put it. How about I go closer? Better. No. Okay, you're okay. I don't know if Charlie will come get me if you can't hear me for the. Oh, I'm getting thumbs up. He can hear me. Good. All right. Yeah, thanks so much, Steve. Yeah, I consider myself very lucky to have been here and in Vermont my entire life, and at the University of Vermont my entire professional career. And I always say about UVM, I think it's really true that nobody really gives you much. There's nobody who's saying like, here you go, here's your next step, but no one also gets in your way. So if you have good ideas and you're willing to do a lot of work, you're able to kind of create your own way. And we also have just incredible collaborators. I'm often surprised that I'll like have some grant idea and I'll Google to find out like who in the field knows this, and they happen to be across campus. And you make a phone call next, you're having lunch and you're, writing grants in new directions together. So, I hope one thing you'll just take away is that lesson of like, yeah, reach out and talk to people, and most of your ideas are actually doable if you're willing to like, build that pathway. And so, yeah, I'm going to tell you about two large cohort studies that I'm involved in. So as Steve mentioned, Dr. Garavan and I run the Vermont site for the Adolescent Brain Cognitive Development Study. That's a 10-year longitudinal study. Adolescents enrolled at age 9 and 10. We'll follow them till they're 20. We're in our 6th year of data collection right now, so kind of give you some overview on that. And the second one is the HBCD study, and Sarah Heil's an important contributor there. That's Healthy Brain Child Development Study, a large longitudinal cohort study of pregnant women enrolled in their 2nd trimester. Will follow until their babies are 10 years old, with the idea being at the end of the day, we'll have a sense of what normal brain and behavioral development looks like from birth until age 20. And that really was the kind of goal for those. And through the competitive grant process, we were awarded a data collection site for HBCD as well. And we're just getting that up and going. And so the way we'll structure then today's talk is I'm going to tell you about the ABCD study, kind of an overview of what that is, with a focus on the fact that ABCD is done with open science. And so all of the data in ABCD is freely publicly available to anyone who gets a data use agreement. So I'm hoping to, inspire a little bit of interest. It's a great source of a huge data set for something that could be the basis of a paper, can be preliminary data for a grant, can just help explore connections between things. And we are happy to help people who are interested in doing that and let you know kind of what's in that data set. We'll give you an update on where we are in terms of data collection, what we're seeing in that cohort today. I'm going to give you a few results. It was hard to balance not wanting to just give you 100 science talks, which might be my preference, but I want to give you the big overview. I'll tell you a little bit about some of the work we're doing, highlighting some of the work we're doing here at UVM, and then tell you about the development of this baby study that we're doing, give you an overview of how that will work, and let you know where that is out at and how you'll be able to access and use that data if you want. So the ABCD study is really entirely sits on the shoulders of Nora Volkow. Nora Volkow is the director of the National Institute of Drug Abuse. And this was really, Nora many years ago just really felt like we have all of this data from small groups of substance users and from some really nice longitudinal work and experimental work. But what we're missing is any normal reference population. So if you're looking at brain changes among, 50 people who are addicted to a drug, we don't have a great set to know what the normal variability looks like. So you can pick 50 controls, but these small studies have problems, which I'm sure you know about and which we'll talk about in terms of the reliability of those findings. And so she felt like, what we really need is a large longitudinal study that's going to start before kids are using substances to help us really understand the normal variability in brain and cognitive development, and to begin to understand all the different factors that go into somebody's trajectory. And so that was the kind of impetus for starting the ABCD study. The ABCD study is, like I said, 10-year longitudinal study. We recruited in 2016 and 2017. Across the country, there's 11,878 children and their families participating. We have an intentional twin design, so 2,000 of the kids are twins, and so you're able to do some interesting, twin research in there if you want. Everyone was enrolled between ages 9 and 10. We're in contact with families every 6 months. So you do on your mid-year, you'll do a phone call, and then on an annual visit, you'll do a whole battery of assessments. And every other year, we do a full multimodal neuroimaging battery, as well as all the biosamples. So I'll tell you more details about that. But it's a huge amount of work. I can tell you that. So in Vermont, we have 575 families. When Hugh and I wrote the grant, we enrolled, 9 and 10-year-olds through schools. And when we wrote the grant, it was 10% of all 9 and 10-year-olds in the state. And I was like, sure, well, let's say we can do that. Let's try that. And we were then, we're amazed that, yeah, we actually were completely able to recruit and to maintain this cohort. So it's a huge effort and a real, I think Vermont is a pretty friendly place to do research. People are interested, people are willing to volunteer their time, people are really tied into their communities, and that served us really well. You'll see, so the flags on the graph there are where all the sites are in the country. In California, you'll see that they have some extra flags. They host the administrative core and the data analytic core for the study. Hugh is one of the 4 associate directors for the national consortium, and he and I both lead a number of different work groups, which I'll tell you about as we go through. So we're very involved in the study, kind of at all different levels, at the leadership level, at the site level, and at these work group levels. Recruitment was done using an epidemiologically informed approach. And so what we did is hit the target demographics were based on the census on the American Community Survey of 9 and 10-year-olds at that time. And schools were selected who had that population. And then every child in the school who was 9 and 10 was invited to participate. So it's not truly a representative sample because you had to be close to a study site in order to be offered participation. But then we tried to do a good job of offering participation broadly. We didn't select any particular demographics to enroll. We did constant monitoring of the sample we were recruiting, and then could tweak that by the schools that you were allowed to recruit from. So you'd be released kind of certain schools to recruit from. And in this way, we're able to kind of keep each site diverse and each site recruiting the demographics. So for example, here we had to work hard to recruit rural participants. It's easy to be in your rural state and recruit in Chittenden County where many people don't meet the definition of rural. And so those were kind of the ways they were like, no, you need to go recruit from these other schools. And so that's how recruitment was done. Twins were recruited through birth registries at special sites that were twin sites, although every site in the consortium enrolled some twins just by happenstance. So our goal was to try to reflect the US population broadly. And so with the 11,878 families, you can see here in blue, this is household size with the American Community Survey here and ABCD there. We've matched pretty well for that demographic, an equal number of male and female participants. Here's our racial and ethnic breakdown, and the way these circle graphs work, the inside circle are the targets, so that's the American Community Survey targets. The outside circle is what ABCD has, what the final sample looked like. You can see we have a 96% match with the national demographics. And just so you see how the graph works, so like in the other race category, the target was 5%. We enrolled 11%. And in doing some qualitative work, trying to understand what that was about, people who are multiracial, we didn't have other options on the way that we initially assessed race and ethnicity. So I think we have a lot of people with multiracial identities in that group. And so we're kind of working on collecting additional data to do some recoding as we learn more about best practices for those types of demographics. The one place that we have matched probably the worst, I just feel like full disclosure should talk about, is household income. And our sample has a higher percentage of people who make over $100,000 a year and a lower proportion of people who make less than $50,000. We think about this a lot because we wanted to do as close a match as we can. However, there's still many thousands of people in every category. So it's not like there are no people of the lowest income, but it is something that we tried to balance out, and this is where we fell off the farthest in that goal. ABCD is when it was awarded, it was the biggest grant NIH had ever given. I think HBCD has actually eclipsed that, but around $300 million is the estimate of what this will cost. So it's a huge investment it's a huge investment of research dollars that other people won't have for their RR1s and those things. So there's a huge sense of responsibility, I think, of doing that. But really, that led with Nora's leadership, the collaborative research on addiction group felt like this was the right decision, and they really led the charge. However, as soon as ABCD kind of became a reality, the site got going, we started recruiting. Many other federal partners came on board and have contributed money to the effort and kind of want to be a part of that. We've had an interesting, there's an interesting balance as a work group leader to you want people's money and support, but you need to have a protocol that still is doable and that people can do. So we have to have processes as a consortium to take new proposals that come and decide what things do you want to add or don't want to add. For example, the National Institute of Justice has funded actually a big sub-study. So a subset of sites who are particularly interested have participated in the National Institute of Justice sub-study, and those and participants at their site are offered, they don't have to, but are offered to do 2 extra hours of assessment that is specifically on factors related to juvenile delinquency and criminality and those things. So that was kind of a balance where we felt like for the whole consortium, it's too big an ask because the visits are already pretty long. And so here are the domains, but I'll tell you the highlight is on the long visits for the every other year, when you're doing your neuroimaging year, our participants are here for about 8 hours. About 8 hours of assessments. They are now high school students, so trying to find 8 hours after school, in the evening, on weekends, is getting harder and harder, but it's always been pretty challenging. The years that are the short visits are still about 5 hours of assessments, and so this is a really huge effort for people. It's a big ask. Someone calculated at our baseline when we released our baseline data without counting any of the neuroimaging variables or any of the genetic or any of the biospecimen variables. There's 44,000 variables released on every participant at baseline. And we are now in we're releasing our 5-year data. So it's a humongous data set. And encompasses things like physical health variables, looking at pubertal development. We do, height and weight, vision screening, those types of things. We get biospecimens, kind of 3 main categories to get pubertal hormones, to get genetic information, and then finally to get substance exposure information. And we get that both in real time when you show up for a visit, kind of the short-term assays, as well as hair samples for longer-term assays. We've collected baby teeth from all our people. Those are actually being analyzed right now through somebody's RO1 that they got to analyze lead levels in baby teeth and map that to some of the exposure maps throughout the country. We have a mental health work group. I've chaired that for many years, along with Deanna Barch at WashU. We do both DSM and dimensional measures of all different types of psychopathology, and with a balance of getting information from parents and getting information from kids, and that's starting to shift, which is appropriate, where we're getting more information from youth as they're now in their teenage and less from parents. A neurocognition battery that's task-based, so we're actually having kids do NIH toolbox tasks. So this group particularly really interested in that. We have a delay discounting measure, we have response inhibition measures, we have good working memory measures, things that are linked to substance use trajectories. A very active culture and environment work group that's trying to look at environment in your family, in your school, in your neighborhood. They work very closely with what's listed here as other data sources, has been renamed to the linked external data set work group. And we've managed to take all of our participants and through geocoding, link to data sets that like the National Crime Database and pollution databases. And it's kind of amazing actually what is available. There's a whole database that has like neighborhood resources. There's databases that look at laws against gender diversity or support for Black Lives Matter protests and the way that money is spent in the state. You actually can triangulate some pretty interesting data about our participants. And of course, a big focus on our substance use battery, again, using both interview-based and timeline follow back on everyone, as well as many other types of questionnaires. And substance use is the one thing that is measured every 6 months. So on the phone interviews, it is primarily to get onset of substance use, so we can try to, as close as possible, pin down when youth are starting to use. And then the thing that probably, the biggest advance for ABCD is this open science model. And this was really so different than any other research I had ever done as we first started doing this, to realize that as we collect data, the data will be made publicly available to the scientific community. And in fact, as an investigator, I cannot publish anything that you don't have access to. I can't analyze anything that you don't have access to. There is no advantage, and in fact, perhaps a disadvantage of collecting all the data, right? Because you're busy collecting it all, but it actually is released to the public. And this, I will say, initially, this caused us a lot of anxiety within the consortium and just in general of like, it's we're in a competitive field, we're all trying to do things, and we felt like everybody's going to be trying to answer the same questions, and how are we going to get through this and collaborate? And it turned out that, of course, there's a million questions, and my most interesting question is not your most interesting questions. And there's also so many different ways to answer a question with all the data we have. So a lot of our anxiety has not borne out, but we have managed, and I'll show you some slides in a moment, to really make good on the promise of disseminating this data and having it be widely used. And for me, there's a really important kind of social justice piece in this, which is people from under-resourced areas, people who don't have state-of-the-art MRI scanners, they get the raw neuroimaging data that I have within a month of it being collected. So if somebody wants to download these free pipelines that are available, and the code is all available on our site, there is no advantage that I have other than the intellectual environment I have around me. But everyone has absolutely equal access to the data. And I think that that's, we see that now, the mandates more and more to have open data and data sharing. And this was really a really stark contrast when it began, and has turned out to really be, I think, a very good thing. So where are we right now? Right now, we're collecting 6-year follow-up data. Our participants are age 15 and 16. And I always can hardly believe it when I tell you that we have a 97.9% retention rate at 6 years. Like, it is crazy. During the pandemic, we did remote data collection. So we are missing some neuroimaging data, some biospecimen data. And of course, around the country, the timing of when places were opened or closed changed. But we were able to collect most of our data and stay in touch with our participants. Now we do have, and so the total, in total, 253 participants have withdrawn to date. And you can't see any of the text down here, but these are every assessments every 6 months. So you can just kind of see where we've lost people along the way, although we haven't lost very many. Now, we don't drop people if you miss a visit. So how many people are there, what we kind of call their de facto withdrawals? Are there people who aren't really engaged anymore? And we're kind of doing our analysis of that now. But in some ways, it almost doesn't matter, because if you gave full data at baseline, which was a requirement to be enrolled, even if you missed the next 4 years of data collection and then contributed, it would be really valuable data. So, our retention is really high. Most of our people are seen at most visits. There's probably a little bit of mushiness in there with people who've missed some visits, but still is pretty amazing. And why is it so good? I think it's so good for a couple of reasons. One is because it's such a big investment to be in the study, I think people are committed. They made that upfront cost of spending that whole long day with us. I hear from parents it's become like kind of a tradition in their family. It's something they do with their kid every year together. We also do a lot of things to try to build that kind of esprit de corps. So here at UVM, we go to Lake Monsters games every year. And I show these pictures for a couple of reasons. First is, if you look at that top picture, these are some of the UVM cohort at age 9 and 10. And just looking at the diversity in development. Some kids are big, some kids are little, right? Some kids, all the different ways they are. And here's a subset of our kids 2 years later, and anyone who's raised kids or has siblings know there's a real difference between 9 and 11-year-olds and 11 and 12-year-olds. And they, you can really see that development. But one thing that is interesting is we see our t-shirts all over the place. If I'm wearing my ABCD sweatshirt, people, approach, I'm in that study and I was a part of that. And I think the school recruitment really helped our consortium because their friends are in the study, their cohorts are part of the study together. So it's been a very fun piece of ABCD. We also have the best staff. They are so much fun, and I like to spend time with them. So do our people. Here's a little bit of the data on how we're doing in terms of releasing our data. So over the last few years shows you the number of grants that have been funded for secondary analysis of ABCD data. And NIH and NIDA particularly always has a call out for leveraging this data for new research ideas, funding both the R series research grants and also training grants quite extensively from this. If you look at grant mechanisms, all of the mechanisms are using ABCD data. The big green there is R01s, which we fund the most of, but a lot of F awards and K awards using ABCD data, feeling like this is a great set of data to do some training and then go collect your own data after hypothesis development in the ABCD. The other thing that's a really nice surprise is it's not all NIDA. So NIDA is in this yellowish brown, but NIMH is the biggest funder of other grants in the orange there. And then you have funding from all kinds of institutions who have, are able to see the value in ABCD and wanting to leverage that. Publications that they're more and more. It was kind of slow start of like, are people using the data? Now there are tons of publications. And on the left, what I'm showing you is the blue bar. The blue on the bar is people who are within the consortium. And the orange is people who have no affiliation to ABCD or an ABCD site. And so we just, again, feel like we're really making good on that promise to get the data out there and enable people to use that. The word cloud is from about 6 months ago of the most common terms that were being published. And this has also been a pretty fun surprise for us. And Gaia Dowling at NIH put this together. What are people publishing about? All kinds of things. Again, we thought we might be competing for ground and that we'd all be interested in the same things. And yet the ABCD data set is so broad that people are publishing in lots of different fields. We've also, as ABCD was running and has been really pretty stable and our cohorts are doing well, we were able to do things like during the COVID-19 pandemic, we wrote a supplement and got money to do some extra measurements specific to COVID-related stress and COVID-related coping, and to send a separate wave of surveys to all of our participants. You see some COVID data there. Again, during the height of the Black Lives Matter protests, and also when there started to be protests against mask mandates, the group at Yale led an effort to add in some measures to find out what are the effects of participating in that type of activism, and are they different for kids of different racial and ethnic groups or different types of protests. And they've published some nice work on civic engagement. So something we never set out to study, but these opportunities are really present throughout here. We're right now a part of the big Recover effort. I don't know if you've heard about that. Recover is a big COVID NIH initiative to find out, really trying to look at like, what are the effects of long COVID, who gets long COVID and that. And ABCD was this perfect place for them because we have 11,000 kids with brain imaging and cognitive data and mental health long before there was COVID-19. So all of our participants are offered the option to participate in the Recover study, in which they do a blood draw to look for COVID antibodies, and then they will contribute the data we're already collecting to see if are there any COVID-related changes that we'll see. So already this is a lot, but I'm going to take you through some kind of key things that we're learning from ABCD. I'm going to highlight some of the work we've done here at UVM, and particularly I want to highlight our START program. It's called the Scientific Training in Addiction Research Techniques, and that's a program that Hugh and I collaborated with a colleague at University of South Florida, Micah Johnson, and Micah has a lot of, he's an addictions researcher with a real expertise in mentoring underrepresented students. And so the START program recruits 12 scholars at graduate students, postdocs, and faculty and early career faculty who are all underrepresented minority scholars, and train them in using the ABC data. So they come for a week to Vermont. They watch our research assistants collect data. We talk about running a lab and do that type of mentoring. Then we go through all of the measures that are available in ABCD. scholars develop a project. Each one is paired with a mentor who is an ABCD investigator who really knows their domain and their area. They work for a year on a project, and the goal is a paper at the end of a year. It's been, again, one of the more fun things that we've done. We plan to go into this area, but it really has been a great thing. So people often ask, what's the biggest thing you've learned in ABCD? And probably the paper that got the biggest splash and the most attention was this paper by Scott Merrick and Brendan Turbo-Clemens, published in Nature last year. And the headline here was that reproducible brain-wide association studies require thousands of individuals. So I'm showing you this graph with a lot of data on it. I'll draw your attention to this third line down here. And what that is showing is, with sample size on the x-axis, the amount of variability you're going to find in results that varies as a function of sample size. So if you have 25 subjects and the true relationship is a zero correlation, you're going to have these huge variability in your study. And at 200 subjects, you're still going to have a considerable amount of variability as you studies to hone in on the reliable finding. And it's not until you get to the thousands that you'll start to get pretty consistent findings. And that's true if you're linking brain structure. So on the far left here, we have cortical thickness linking to cognitive ability or psychopathology. It's also true for studies using resting state functional connectivity. And that is a way of measuring brain network activity. And it's become very, very popular and is used a lot. However, it suffers from the same small effect sizes problem. So, sometimes I joke we spent all this money to learn that effect sizes are small in our field, but I think you can have that as the kind of disappointing finding, or you can take it as a challenge, right? We need to improve our measurement. We need to improve our methods in order to be able to get these findings. And we need to rely on our large studies to develop hypotheses to then take into experimental research and have a better chance of it being grounded in fact. Another study along the same lines was done by Max Owens when he was here. He's now a faculty at McMaster University. And what Max did was he took the non-imaging data, so all the behavioral data in ABCD, and summarized all the different domains to 195 summary scores. So a summary score for cognition, we had, the height and weight scores, you have your different mental health scores, and took those and did just a massive correlation matrix, right? To see what are the size of the effects. And so things like, your fluid intelligence and your crystallized intelligence. So two different kinds of intelligence, about how highly do you think those are correlated? Or your age and your height, how highly do you think those are correlated? Or your impulsivity ratings and your mental health diagnoses. So you can take a second and just guess where you think they're going to be. And I'll flash them up and give you a minute to read through some of these and see. And kind of the takeaway here is that in the top, you see some things are fairly well correlated, right? So a correlation between sleep and mental health problems of 0.58, pretty high correlation. Fluid and crystallized intelligence is at 0.48. I'm surprised it's not higher. They're from the same types of cognitive tests. We know 2 forms of IQ should be, but still that's a pretty strong correlation. Parents report of how your kid is doing at school and their CBs and their attention problems pretty high. But most things are much smaller correlations. So the parent reported how many traumatic experiences your kid has had, and their total psychological problems correlates at 0.2. The UPPS is a measure of impulsivity, so the lack of planning score, and your attention problems on another rating scale, 0.17. And then down here, even smaller correlations. So that lack of planning and how many detentions you get is 0.08. Sleep problems and IQ, 0.06. Physical activity and weight only correlates at 0.03. So we have a lot of very small correlations. He was looking at those, and the summary kind of here is that the median correlation is 0.03 from all of these types of measures. So when you wonder, why do we have trouble finding consistent associations? These effect sizes are really small. They're a little bit bigger if you look within a specific measure, if you look within a specific domain, so different mental health measures correlated with each other or within a specific reporter. But essentially they're all these very small correlations. And he looked even farther to say, like, well, what if you only took significant correlations and we tried to see, are your effects bigger then? Or if you really were careful about selecting regressors and tried to regress out everything you could. And basically they're so across neuroimaging and non-neuroimaging data, what we find is that you have small effects and you need big data in order to be able to find any of these associations that will last. So that was kind of our initial set of findings from ABCD. And I think that led us to think about, like, so these large cohort studies then become pretty critical, because it's not until you get over these thousands of people that you have really reliable associations. It says we should spend more time on our phenotyping. I think I'm probably preaching to the choir here, but that if you're not really carefully thinking about how you're grouping people and how you're conceptualizing people's behavior, you're just adding a lot more noise in. And I think something like ABCD allows you to use multiple different measures to try to build more robust phenotypes. With the large cohort studies, we can do much more robust analytic methods. So a lot of the work we do now has built-in replication studies, or you'll take a subset and do model building and test your model, and the other half of the data or another piece of the data that wasn't used for training to really try to avoid model fitting. I think that's been really helpful for us. Beginning to be able to disentangle the confounded demographics. You also can do a lot of different types of methods, and I'll show you a few of them where you can take all of the data in ABCD and you can define a phenotype of interest, and you'll be able to select a matched control group from other participants. And so that, I think, has also really increased our power in these types of studies. So the first example of a specific finding I'm going to give you is from Priscilla Dibbs Gonzalez. She's one of our START scholars, and so our ABCD subjects are not using many substances yet. They still are very early experimentation with substances at age, this is up to 13 here. And so she was like, okay, I'm really interested in substances. I can't really study that yet. So look at family history, because we know that family history has a strong relationship to later use. And she was interested in brain development. So is the trajectory of brain development from ages 9 to 13 different in kids with a positive versus negative family history of alcohol problems? On the bottom left, you'll see how she defined the group. So positive family history was at least one parent and or both grandparents who had substance use problems. Family history negative was no parents or grandparents with problems. And then she left out the group in the middle, and that's another analysis we could do. So there were 1,500 kids who had one parent or one grandparent who had substance use history. So it kind of made these groups. And what she found is that between ages from age 9 to 13, between age 9 and 13, what you'll see is that in the frontal regions, there's significant differences in the positive group, and they have more rapid thinning. So what's going on at the brain in ages 19, 13 is everyone is having cortical thinning, right? Through the pruning process, that's the normal process. Those with a positive family history are having accelerated cortical thinning in these frontal areas. And so this could then serve as a marker of vulnerability. You can then, her next step is taking these brain areas and now searching backwards for, are there other cognitive, behavioral, social markers that may help us find areas to intervene on, and that may help us identify this vulnerability. Another START project from Faith Adams, I wanted to talk about. So again, kids aren't drinking much alcohol, but we can look at the alcohol expectancies, and there's a couple of items from that. So a positive alcohol expectancy are your belief that something good will happen from using alcohol. So it will help you relax, feel happier, less tense. It can help you get along with others, those types of things. And so we're asking kids who are not drinking yet what they think drinking is going to be like. And what we know is that these positive expectancies are related to earlier alcohol initiation. They're related to heavier substance use later on. And the idea here is that these personal beliefs are probably pretty modifiable with information. And if we can help figure out who has these positive alcohol expectancies and kind of where they come from, we may be able to have a place to intervene. She was also interested in, do these positive expectancies relate to brain function? And does this also help to explain some of the broader phenotypes that we see with kids who are vulnerable to addiction? And so she chose a form of impulsivity called response inhibition. It's the ability to stop a prepotent response. We often think about that in addiction, like if you are a cigarette smoker, whoops, I didn't take you there yet. If you are a cigarette smoker and you see a cigarette, people will often say, you know, they're smoking before they even thought about whether they want to. They lit up before they even had a chance to kind of process that. And that's that response inhibition. Like, how much time does it take you to weigh the consequences of your action? And so what she found is that the people with the higher expectancies were more impulsive on this measure. So they had it took them longer to stop themselves from doing an action they had started. And that was related to less recruitment of the anterior insula during the response inhibition task. And so she's trying to kind of triangulate these expectancy beliefs to a type of brain function that you may be able to then get a cognitive treatment that will help with both processing consequences of actions, feeding back to more realistic alcohol expectancies. She's a grad student down at Harvard, so she's young and early in her career. So be kind to her when you see her at conferences. She's going to do great things. She's been a lot of fun. The last example, I have the last example I'm going to give you is a study we did here with prenatal cannabis exposure. This was, grounded in the fact that we see use of cannabis during pregnancy is just much higher than it's ever been. We know that THC crosses the placenta, and we know it accumulates in fetal tissue. Yet the long-term effects of this exposure, we really don't know. And in 2017, the conclusion was that reduced birth weight was really consistently linked, but nothing else was seen that consistently related to prenatal cannabis use. This is work that was done with Leanne Sessedri, who's a pediatric hospitalist here, and she chose a case control model in ABCD, which I think is a kind of unique opportunity. And what she did was looking at the parents reported their use during pregnancy. So this is 10 years later. They're saying what they did when they were pregnant. She identified 224 people who used cannabis after they knew they were pregnant. You asked, did you use it? And then did you continue to use once you knew you were pregnant? And 224 parents said yes, they did. She then matched on a group that had tobacco and alcohol exposure at the same level without cannabis exposure. Because as you can imagine, the parents who used cannabis also used higher levels of alcohol and tobacco. So here she was able to match on that to try to isolate the effects of cannabis, and then to match on demographics to a non-exposed group who didn't have any alcohol, tobacco, or cannabis. So a nice way to kind of try to isolate those effects. She ran a series of mixed model analyses, and one of the things in all of our research, particularly in these big data sets, is picking your covariates. And so she chose covariates related to ADHD because she was interested in attention problems and wanted to try to isolate those effects, looking at neurodevelopment and also any demographics that warrant equivalent after the matching procedure. She did looked at mental health outcomes and also cognition outcomes, and then the 3 MRI tasks looked at a vertex-wide analysis of that. And what she found is that prenatal cannabis exposure was associated with attention problems and externalizing problems, so like acting out behavior. And that was consistent on parent report and also on the subset who had teacher report. But there were no effects on cognition, no effects on brain activation from any of the tasks. And so we've had a lot of long discussions about how do you get attention problems that are not related to brain function or towards cognition? Like, what is this about? When is this happening? At the same time, actually, right before she submitted hers for publication, she was scooped, as will happen in these studies, with a big paper in JAMA that used a very different method in ABCD, but found pretty much the same things, no brain effects of prenatal cannabis exposure. So really interesting, and yet we see these attention problems. And so studies like this have led to maybe there's a motivation to do another big, large study, because wouldn't it be great if you could get measures of the prenatal environment in real time instead of asking someone 10 years later? Wouldn't it be great if you could measure all of the other environmental factors? And that's really the drive for the HBCD study, is to collect this in real time to get much better measures, including biological measures of the prenatal environment, and helping to untangle prenatal substance use from the postnatal environment. And so, that was the impetus for that. And these studies are completely different groups of investigators, but there are synergies in the protocols built in. And in fact, Hugh and I at the University of Vermont are the only investigators in the country who are the PIs for both sites. So that's kind of a cool, we like it for ourselves, but also for UVM, being able to be a player in both of these large cohort studies. There are other universities that are in both, but they have completely separate investigator teams. And I always think about too, like, what's the motivation to try to really understand the early development? And I'm just going to show you the gratuitous pictures of my own kid, right? So here you go, right? From birth, this is her at age 15. She's now 25, living in a van, skiing, driving around the country, having a heck of a good time. But you think about all the myriad of things that happen, and wouldn't it be amazing to have better advice for parents and better understanding of what was going on during development to guide us? So I'll just tell you briefly, so we have a few minutes for questions, about the HBCD study. So it's a 25-site study. It's about 1.5 times the budget of ABCD because it's even harder to study infants. Sarah Heil has been really critical in our team and the work that Steve has done to prove that Vermont can do this. And Vermont, we have incredible reputation for being able to work with substance-using pregnant women, and that was really commented on by NIDA very positively in our application, and is proving to be very true. This will start at 7,500 mothers. The goal, ABCD started at 7,500, and as recruitment went well, other people kicked in money, so HBCD could get bigger. We will see. 25 sites in the country are involved at this point. Oversampling substance-using pregnant people. So the goal here is that we have external validity to the general population. That's the big bar on the left, and that's what these demographics, taking that in. But that also, we are enhancing to have a demographically diverse group of substance-using pregnant women. Targeting 25% of the sample will use substances during pregnancy, with about 12% of any of opioid, marijuana, alcohol, and tobacco. And of course, we won't have to enhance much to recruit tobacco users. Pregnant women, to get to 12% won't be as hard as it will be to get to 12% of our sample who use opioids during pregnancy. So there'll be selective enhancements to try to get pregnant women using substances at that rate. And we will also be very carefully capturing a group who are demographically identical who don't use substances. And so we have a strong epidemiology team here who has a dashboard that will monitor in real time, and they will tweak the clinics you can recruit from or the number of people that we enroll at birth. We're right now targeting no more than 10% of the people in our study enrolled at birth, but a lot of people who don't get prenatal care, we obviously won't be able to recruit them early. And so that may be something we need to do to both enhance substance use, but also the population that matches substance without using drugs. So that's the plan there. We'll see. We're running pilot studies right now, with the full study targeting to start in April. This will use MRI, similar to ABCD. We'll also use EEG for babies. So we've learned how to do that. It's been kind of fun. The same large assessment battery with biospecimens, substance use, environmental measures, also doing wearable biosensors, both for the mom and for the baby, collect that type of data. Again, broad support through NIH for this. You'll see it's healthy brain child development with the HEAL capitalized because a lot of the money came from the HEAL initiative to begin this study. So thanks to them. I won't even go through that, but I will say we have a huge kind of leadership structure. Hugh, again, is part of the, is one of the 4 associate directors at the coordinating center. I chair the neurocognition and language work group for this study. We have 12 different assessment work groups all working hard to do that. And I'll just say it's been really fun. It's a scientifically much more diverse group of investigators. So ABCD, we were all adolescent substance use researchers, and we had that in common. HBCD is a much more diverse group of pediatricians, OBGYNs, substance use researchers, and it's been a bigger challenge, but I think it makes for a much better study to have kind of come together to try to design this. All right, that was a lot of talking. In summary, ABCD studies really going well. That data is just sitting there waiting for people to analyze, and I am happy to talk with anyone who has questions. I actually, as part of our START program, I put together a really nice PowerPoint that will give you every measure that we have in every domain. So it's kind of like, I wonder if you had a measure of X, Y, or Z, shoot me an email and I'll send you that, that PowerPoint. You can look at that. The open science has been really fun to watch and is really working. There are now some data analysis tools that you can just log on to once you get your data use agreement. You can log on to, it's called DEEP, the Data Exploration and Analysis Portal, and you can actually run statistical models right from there using the proper families nested within sites. If you have the twins, you have to account for sibling relatedness, and that can do some of the machine learning replication studies. It's all it's been a very cool advancement, and really hopeful that these 2 studies together will really help us understand normal development. And just a huge shout out to, this is the HBCD team at UVM here on the right, the research team that runs ABCD there on the left. You'll see us often in our t-shirts. And I will say, I counted it at 1,400 hours of assessment every month, 12 months a year, going on 60 months. So they're really doing a tremendous amount of work. So it's okay. Thank you. Sure. Oh, I did.
So folks can ask questions using this handheld mic, and folks online can post in the Q&A, and I'll read those off.
I'll also be at lunch for new people who are coming over to lunch.
Thanks. That was a really exciting talk. I just incredible work you're doing here. I didn't know. I knew some peripherally some of this, but not all of it. So great to hear. I have a couple of questions, but I think one most pressing is the first study you showed published in Nature. So is your interpretation that then that the community should not be conducting neuroimaging studies with under 1,000 patients? Like, is this the future of neuroimaging that we're looking at?
It's a good question. So that question being like, you have to have 1,000, like, can you just not do it if you don't? I think the answer is no, but I think you shouldn't do a population study with 50 people in neuroimaging. I think that we have to get much better at asking much more specific questions. And, I used to joke when I started grad school, and it feels like it wasn't that long ago, although I guess it was now, but, you could get a dissertation by saying, we'll put it in the scanner. Like, what does OCD look like? I know, I'll do an imaging study of OCD, because we had no idea. And, we can't do those types of studies. Those are not going to help us. But you can do studies of, that really mixing good cognitive neuroscience. So getting using a data set or using some behavioral data or an MTurk study to isolate a cognitive process, that in a scanner with 50 people will give you reliable data. It's really when you're trying to link a brain to a phenotype from a big sample that you need more of. I also think, and I'm excited to hopefully get back to my old roots at doing these within subject studies, doing pharmacological challenges, doing studies of neuroimaging studies of people during treatment, during abstinence. Those studies have tremendous power, right? Because you have a within subject design that are, carefully selected. Again, you probably need to be really, really thoughtful in the phenotype that you're doing, the level of addiction, the type of person. So I think that like the days of phishing studies are a huge waste of money, but that really, how you're all trained, good behavioral pharmacology study can still really benefit from them.
So I don't think that you haven't tested this, but I was wondering, do you have any idea why attentional processes would be affected by cannabis exposure, like prenatally, and cognitive processes wouldn't be?
Yeah. So
Idea?
yeah, I thought a lot about this. I think it's a great question, and I think that it actually kind of relates to some of Eli's question. I think that so attentional problems that are reported on a self-report represent a myriad of cognitive underlying problems, right? So that you can have trouble interrupting someone. And that could be because of working memory, where you're unable to kind of keep your mind on the conversations you do. It could be impulsivity. You can't stop yourself from doing it. Could be emotion dysregulation, where you're trying to interrupt somebody who's saying something you don't like to hear. Like there's so many different processes. And so, my guess is, we know that ADHD you can get to by, something like 5,000 combinations of symptoms. And what I think is that those that our DSM diagnoses don't work for that type of study. And that if we can narrow in on what is the attention deficit that's linked to cannabis, and I think we don't know yet. I think we haven't, it's not fine grained enough yet. I think it's probably very variable. Again, depending on all of these factors, we're getting these kids at age 10. So lots has happened in their brain, right? Lots of adaptations, coping skills, and those things. And so the other hope is that HBCD, when you can do like EEG and just see signal processing in a 3-month-old, hopefully that'll be much more linked to these core processes. That's a great question.
We have a remote question from Mariah Flynn. Are there researchers connected to the ABCD study that are able to give community level presentations or workshops on the results of the study to connect the research and information you are gathering to more of the general public, particularly parents? I've been following the results, and it is all really helpful information for folks who are working with and raising kids right now.
Yeah, absolutely. Thanks for that question. We do an annual presentation here through UVM, although it's been online the last couple of years. But feel free to reach out. You and I both like talking about our work in these studies, and we're happy to come to do that. We, through the early recruitment, we did talks at all of the schools and a lot of community lectures. And you're right that as results have come in, we do less of that because we're just busy doing other things, but we would be more than happy to, and we'll try to make sure we broadly advertise our annual updates.
A question about getting involved with an initiative like this from just sort of a professional development perspective. So, with all the sites across the country for each of these two big studies, was there like a call for applications? Are these colleagues that knew each other already, or how does one become involved with an initiative?
Yeah, it's a great question. So both ABCD and HBCD use the same process, and NIH put out a call for applications, and you could go in as a consortium with other sites. You could go in alone as a standalone site, depending on what you thought your strengths were. ABCD, I think what we know now is that there were 10 different consortia that put in applications. We were part of a consortia. Our coordinating core and data center were funded, and then about half of our original sites were funded. And then the other sites were drawn from other consortia that weren't funded but had a good application, or from independent people. So it was interesting, and it was a pretty, as you can imagine, challenging for people who wrote our grant with us who are not ABCD data collection sites. And so, I will say, if you have a good, if you're, here in Vermont, we have a really unique population that can contribute to these studies. And so even if you're not part of a consortia, you can still go in as that standalone site, and will often get added to a consortia with a good application. I've been surprised at how successful that is for some some of my colleagues. And when ABCD started, I was, I think, the only PI who was a assistant professor, but there were two very new associate professors who have standalone applications and weren't part of a consortium. The other thing, and I think this is just general professional advice, is, let yourself say yes. When Hugh came to me and said, hey, I think I'm going to apply for ABCD, are you interested in doing it with me? I said no, because I had an R01 that was almost funded. I was in the fourth year of my K award. I was running down, things and wanted to do experimental psychopharmacology. And if my R01 had been funded, I probably wouldn't be doing ABCD. And as time went by, and I was like, why would I say no to this opportunity? And luckily I said, yes, I will do that with you. And recognizing that it meant putting on hold some of my other things, right? Recruiting this sample and getting this study going was a big investment. And man, the best investment I've made in my career. That's the colleagues I have, the amount of the work I've been able to do, but you don't know. So I do say, you have to take some risks. And if you're going to have to make those choices and go with them, and it's hard, because we'd all like to be able to be 100% successful in our first choice, and that may not be the way it works out.
And so that wraps it up for the remote audience.